Okay, so Lawrence and Fred asked me to give a talk on kind of genomics and how it's relevant to deep history in Africa. And I'll confess, it's not really what my lab works on much anymore. It's something that we did when I was a PhD student. So I don't have any big kind of impressive new research to present. So this is why I'm giving you a kind of a review of how the history of how genetics has helped understand African and particular sub-Saharan African pre-history. So you've probably all at some point heard the phrase, there is more genetic diversity in Africa than anywhere else in the world, which is true. However, that in itself is not particularly interesting. The fruit flies have more genetic diversity than humans, right? The reason it's interesting from a population geneticist point of view like myself is that you can then layer that on to the immense amount of other kinds of diversity that you have in the continent. Phenotypic diversity, the ecological diversity. And then the two elements have really framed how population geneticists like me have thought about human African genetic diversity is the variation in subsistence strategies, the farmers and hunter-gatherers, and language, the immense language diversity, and trying to kind of see how that is structured within the context of the genetic data. And people have always been interested in particular in the kind of spread of Bantu-speaking languages and how that is reflected in the genetic data, and also what the genetic data says about click-speaking individuals, hunter-gatherers, and these kind of things. Now, before people could really look at DNA, and like I said, this is an overview, a review, so I'm going to go all the way back to the beginning. We didn't have direct ability to look at DNA. We had to look at so-called classical genetic markers, proxies for the DNA. And this kind of started with Ludwig Hirschfeld in 1919 when he discovered there were blood groups. And in fact, these blood groups, we had A and B, actually had variation in them depending on what populations you looked at. And you can see that he talked about races here and had some certain words that we really shouldn't, we wouldn't deem acceptable. But the principle kind of was that there is variation in populations for blood groups, and that would underlie genetic variation, right? Because DNA makes RNA, makes protein. And over time, stretching all the way to the 70s, people developed more and more assays where they could look at protein variation as a proxy for genetic DNA variation and tried to see if they could use that to understand human population history and the variation that exists. And this is kind of, he's not the only one that did this, but of course the most famous example and the kind of almost culmination of using these classical genetic markers was by Luca Cavalli-Sforza, who in, I think, 84 and then 94, published this very famous work, The History and Geography of Human Genes, which is this kind of incredible effort to assemble all the data that existed and try and come up with ways to explain how people have moved all around the world. And of course, he looked at Africa. And it was, in this case, he looked at 49 African populations. Here's a tree he made. Let's see if this works. Yep, there's this tree he made. And obviously, from the perspective of what we do today, it's a fairly kind of primitive thing, but it does have features in it that would give a kind of little indicator of what we would see in the future when we looked at real DNA. And it kind of identified that there were differences in some of these groups, like hunter-gatherers, Mabuti, groups that were very recently, until recently, very hunter-gatherers, San, the Eastern African individuals from Ethiopia were different to other sub-Saharan African individuals from a classical genetic point of view. And the kind of languages of the Bantu language in particular, individuals from all over kind of the Bantu spread of, across Africa, from South Africa, Southeast, Southwest, North, genetically clustered together, even though this is kind of you would think that it would show more spread given how widely distributed they are. Genetically, they seemed, from classical markers, very, very, very homogeneous. And this allowed Cavalli to kind of present, these kind of models, like using the genetic data from the trees to think about how populations might spread and how the Bantu expansion, what kind of the streams of the Bantu expansion might exist through Eastern and Western and which one came first. And obviously, there's a lot of simplifications here, but it was kind of a first attempt to really do that, to model migration with genetic data, and also to think about things not in a tree-like manner necessarily, and this is something that's become very important in the modern genomic era, but to think of that there is actually populations that are the result of admixture process, of mixing of two populations coming together and creating a third. And this idea, for example, the San and the Ethiopians may have been a result of mixtures of different types of other people, in this case, sub-Saharan African and Near Eastern. In one case, this is probably somewhat correct, and in the other case, probably not. It was limited data, but it was kind of a starting point for how everyone would really think about contextualizing genetic data. It hasn't actually changed that much in some ways. It wasn't until really the 80s and 87 that we actually started to look at real DNA, and this is kind of the real kind of signature of this is this very famous paper by Rebecca Kahn in 87, which essentially identified this mitochondrial Eve and had obviously such huge implications in distinguishing between the multi-regional and out of Africa hypothesis. And it was this kind of was the beginning of a kind of a series of papers from in the 90s and kind of early 2000s that were dominated by genetic work looking at uniparental markers, the mitochondria and the Y chromosome. And this was driven by essentially the kind of ability to look at restriction fragment length polymorphisms, places in DNA that we necessarily couldn't sequence, but we could tell that they were different between people, and also the development of PCR, which allowed us to actually go and get bits of DNA we were interested in, amplify them and examine them as much as we can. And that really revolutionized molecular biology in so many ways. Right. So like I said, the kind of 90s and early 2000s was really the Y chromosome mitochondrial era. If you wanted, you could call it the genographic era, the National Genographic era, because that really is a kind of Spencer Wells was kind of running around telling people who they really were with their Y chromosomes and their mitochondrial DNA. And that became a kind of a real, kind of a precursor for like 23andMe, where you would actually go and send your DNA to them and they would tell you your Y chromosome and mitochondria. It became very popularized. They were actually sending it here to the University of Arizona with my old postdoc advisor, Mike Hammer. And so when you're looking at Y and mitochondria, though, it's important to understand what you're looking at in terms of history, right? So how do we use genetic data to understand history? Now, fundamentally, we don't actually care about DNA. What we care about is for any particular individual or set of individuals, the genealogy back in the past. And if we can work out what that genealogy is, we understand that genealogy of all our relatives is shaped by our history, where we've moved, who we've encountered in the past. So we want to get at this genealogy, and DNA is essentially a kind of fuzzy lens of looking at that genealogy. Now, when we only look at the Y chromosome or the mitochondrial DNA, we only get a very limited view of that genealogy. So if we're looking at the Y chromosome, for example, of this guy here, what we're really looking at is DNA that comes from ancestors along a purely paternal line. When we're looking at mitochondrial DNA, along a purely maternal line. What we're not doing is looking at all the other ancestors that have contributed to someone's history, all their different ancestors. And of course, that's very important, right? There's obviously reasons why there might be interest in just purely male and female lines, but this is a huge number of other individuals, and they all contain the kind of remnants of our history. Doesn't mean that Y and mtDNA is not important. It showed some very broad kind of patterns that were not so different to what Cavalli showed. So for example, here is a principal component analysis of populations of the mitochondria, and it showed that Eastern Africans were different to Southeastern Africans, but then there was a kind of a Central Africans and West Africans. You actually couldn't really differentiate that well through mitochondria. You would see similar things in the Y chromosome, where you would see that there were actually Y chromosome types that were found in Eastern Africans, like from Ethiopia and from Click-speaking Khoisan, that weren't found in Niger Kordofanian speakers from West Africa and Central Africa. And so you would see these kind of deep variations between populations. And the way this one in particular has always been explained, or kind of, is that there's this deep connection between Ethiopians and Khoisan and other click-speaking languages like speakers like Sandari and Hadza, where they contain parts, Y chromosomes that are in the basal parts of the tree, the deepest part of the tree, whatever you want to describe it. And it's completely absent from the majority of people that live in Africa, the Niger Kordofanian speakers, Niger Congo speakers. And maybe this is because at one point in time, this kind of ancient primordial population was spread much more widely across the continent, across Eastern Africa, across southeastern, southwestern Africa. But then at some point, it gets erased by Bantu speakers coming along and kind of leaving these little pockets of these ancient Y chromosomes around, which is an interesting idea. However, you have to be very careful about interpreting Y chromosome mitochondria. It is just one lineage along the human tree. And indeed, while this kind of deep clade was seen as meaningful back then, some years later, we found that actually the deepest Y chromosome clade, A00, which was 300,000 years more diverged than from any other Y chromosome that we had found, was found in the Mabo in Cameroon, in the grassfields area. And no one would say that the Mabo are some kind of remnant of an ancient African hunter-gatherer population. It's just the fact that sometimes the Y chromosome, you're looking at one lineage, you get random results. It's a kind of a random thing. So you have to be very careful about just looking at these single locus because they lack a lot of power. They also lack a lot of resolution. So when I was doing my PhD, we collected a lot of samples from Cameroon and the Cross River region because we were interested in Bantu expansion, and from Ghana. We had hundreds and hundreds of samples from individual towns. And when we looked at the mitochondria and the Y chromosome, we really didn't find a lot of genetic differentiation with the Y and mtDNA. We didn't see any real correlates with genetic distance. So here's a pre-CA, and each dot is a different cross-river Nigerian population, but you can see they're intermingled amongst South African Bantu, Rwandans. Doesn't seem to be a lot of genetic differences there based on geography. And similarly, we didn't see a lot of difference based on language. There wasn't any correlation between language and genetic diversity. And this is not because it doesn't exist, but it's just the limitation of just looking at Y and mtDNA. Because last year, we were able to look at these same populations using a whole genome approach. And by doing that, we can now see, actually, we can distinguish individual towns at the genetic level, that we couldn't see that information with the Y and mtDNA. We could only do it by looking at the whole genome. So what is it about the whole genome that gives us this power? So like I said, this is what we would normally look at if we were just looking at Y chromosome and mitochondrial DNA. Now, as we entered the kind of late 2000s and 2010s, we moved into this new genomics era. There were two technical innovations, microarrays that allow us to look at 1 million different positions in the genome simultaneously, and then eventually DNA sequencing. So we could look at most of the DNA across the whole genome and places where people showed variability. Now, if we look at this same individual where we were looking at the Y and the mitochondria, but now we look at some chromosome, like, I don't know, chromosome 4. And so we've got this chromosome 4 here that this individual inherited from their father, and this chromosome 4 here that they inherited from their mother. And then we look at a specific position along that chromosome, say position 1 million or whatever it is. When we're looking at that position, what we can see is there's essentially a lineage that we can see back in time that reflects that particular position. That DNA is that kind of inheritance. When we look at the other chromosome, we can see this inheritance. So now we're not looking along this line, we're looking at this line and we're seeing different ancestors in the past. We're looking at DNA from different ancestors. Then there's this process of recombination, which shuffles DNA during meiosis. So the DNA that you get from your parents is kind of this mixed up version. And so let's say there's a recombination event here in these ancestors, then that switches who we get to see in that part of the DNA. And similarly, we move to another part of the DNA where there's another recombination event, and now we see another set of ancestors. So by looking at the whole genome, we see much more than just this one ancestor here and this one ancestor here. We actually get to see DNA from many, many parts of our ancestry, which obviously gives us this extra power. And you can see the more DNA we can look at, the more of the genome we can look at, the more of these ancestors and the more complete picture of this genealogy we can get, which is ultimately what we want. We want to know, if we can get this whole genealogy, that's exactly what we want. And that provides us a wealth of information. So Sarah Tishkoff's paper in 2009 is kind of the really first big paper that kind of did this at a kind of broad level across Africa. And so this is kind of, hopefully, at this point, you're all familiar with these kind of cluster analyses and admixture analyses, where each line is essentially one individual, and the color represents the genetic ancestry that individual has. So if you see a bunch of lines that are orange, those individuals share a very common ancestry, but individuals can also have a mixture of orange and green because they've got slightly different ancestries. And you'll see that, not unlike what we saw with the mitochondria, Eastern Africa is kind of different to Central Africa and Western Africa. You see this kind of more purple ancestry here versus it's kind of orange. And this kind of pattern actually is not dissimilar to what we've seen when we've moved from kind of SNP arrays to whole genomes, where basically you can see there's a kind of West African, Central African similarity, differences in East Africa, and so on. And this, of course, has allowed people to look at the Bantu expansion as well and look at the root. And it's not unfair to say this looks a lot like the initial Cavalli-Sforza figure, because that's had such an influence. Maybe it's not exactly the same root, but this has basically shaped how we think. That initial figure has really shaped how we think we're going to use genetic data when we kind of look at these analyses. The things that might kind of stand out to you, there are a few populations here that are in Central Africa and Southern Africa that look a little different. We have the Biaka pygmies and Mabuti, and then kind of San and Khoisan populations. They have a different ancestry. Again, something that was hinted at in Cavalli-Sforza's original trees, even if it was kind of looking at it in a different way. And this actually led to me taking some of that data and some other data and seeing not just can we see who's different to who, but can we model the explicit relationships of these individuals. And so I kind of did the first modeling of how San were related to pygmies and Niger Kordofanian populations. And we found that the best model was that San were the most diverged human population amongst Africans and basically amongst humans. And this divergence took place somewhere between 100 and 200,000 years ago, depending on various assumptions you make. And to be honest, though this was kind of based on fairly simplistic data, that this pattern has been replicated a number of times with whole genome data since then. However, I will draw your attention to, this is the model that was favored, this very simplistic model, this tree-like model. And you'll see that later that was kind of a really poor representation of how things really are. But that's kind of how we often think in evolution. I've seen many, many trees from other people here looking at fossils. We think of a tree-like system, and that's how we understand the world. And so what is the problem that you have with autosomal DNA? So genome-wide data has given us a lot, but when I look at this kind of thing, is this the best we can do in terms of understanding human history? All this money that we spend sequencing all these base pairs and collecting all these samples, and we can make these kind of tree-like things, which is nice. It's nice to know the San are the most diverged, but is it really telling us anything really fundamentally important about history, or this very macro level, broad level things? And so part of the problem is even genome-wide data doesn't give us everything if we look at only modern populations. So if I look at this guy here, this is all the individuals that I showed you before that had contributed some DNA to this individual. And you can see when you go to the grandparents, actually there's one grandparent, even though it's clearly their grandparent, didn't contribute any DNA to this person in the future. And if we go all the way back 5 generations, only 25% of the individuals actually get represented in this individual's DNA. The other ones are clearly part of their family, but just by the luck of the game, their DNA doesn't make it into this guy, right? And so this is why history is written by the genealogical victors, because these are the ones that we see today, but we don't get to see any of these other guys who were probably very important. And David Riker, probably as most of you know, has actually kind of had this nice figure in his book that kind of really formalizes this concept. This is just a cartoon, but these are the kind of real estimates. And if I look at myself and I go 15 generations ago, it turns out of all the ancestors that existed 15 generations ago, only 3% of them probably contribute DNA to my genome. 97% of my ancestors never actually contribute any DNA to me, right? So that's a huge amount of that genealogy that we're missing, the past that we're missing, just because we're fixated on having to limit ourselves to looking at my genome or modern genomes. Now, hopefully you see where this is kind of going. Well, why can't we just go and fill in the gaps by just sequencing some of the bones? Why can't we go and take some of these skulls and from Turkana and use our amazing sequencing technology, and now we can fill in some of these gaps here that we couldn't get by looking at modern samples. And of course, when I was doing my PhD, we never thought this was remotely attainable. But the world has completely changed because of this guy here, Svante Pääbo, when he sequenced the Neanderthal genome and rightly received the Nobel Prize last year. And it just has been a kind of just an absolute revelation in terms of how we can now get DNA from ancient material. And indeed, last month, we hit 10,000 ancient genomes, which is quite an achievement. Within 10 years, we've sequenced 10,000 ancient individuals at certain levels. Most of this data, but not everything, is generated by three big groups: David Reich at Harvard, Johannes Krause at the Max Planck in Leipzig, Johannes' student, and S.K. Valislev. Now, this is great, and my lab has been doing a lot of work on ancient DNA in Europeans. And I have like a student that's got 500 genomes they're analyzing as one chapter in their PhD thesis. However, what you will notice down here is Africa. Of these 10,000 genomes, only 3%, very small amount, are actually of African origin. And that is an issue if we want to use ancient DNA to understand history. So why is this a problem? So when we die, the enzymes that protect our genome, they're no longer around. So the DNA starts to break up into little pieces and even changes. We get postmortem damage. Now, we've got very good in the ancient DNA world of sequencing these tiny little bits and even correcting some of these changes. However, the rate that this happens is very different depending on the environment. So the oldest DNA that we've been able to sequence so far is about 1.2 million years, which is a mammoth. It comes from permafrost. So DNA survives really well in dry, cold environments. It does terribly generally in hot, humid environments, which obviously is problematic for getting material from Africa. So that has been a big stumbling block. Any kind of area that's tropical, in particular, is very, very difficult. Now, these are all the genomes that have been sequenced, published. I'm sure there's more. It's hidden in David Reich's lab somewhere. But these are all the kind of genomes that we have from Africa. And you can see there's a lot from Kenya, which is hopefully nice to see. But it's only 166. Like I said, I have a PhD student working on 500. And there are only 13 that are more than 5,000 years old. So that is a huge limitation on what we can do in the ancient DNA world. Right? When we're thinking about Europe and how ancient DNA has transformed our understanding of Europe, we basically got individuals that cover the entire span of when humans have been in Europe. So we have everything, basically. We don't have anything like, obviously, what we want from Africa. That doesn't mean ancient DNA hasn't been useful. So the first ancient genome that was sequenced was in 2016, which was an Ethiopian individual. Well, he wasn't Ethiopian, but he was from where Ethiopia is today, from around 5,000 years old, Motta, the Motta Cave. And kind of interestingly, or maybe uninterestingly, depending how you think about it, when they looked at this individual and who they were genetically most close to amongst modern populations, was most similar to Ethiopians. Okay. But that's interesting in itself because it suggests some level of continuity for 5,000 years, right? That's an interesting finding. But it wasn't exactly the same as well. It lacked some ancestry that modern Eastern Africans have. And it turns out that this ancestry that Motta lacked, that a modern Ethiopian walking around has, is the same genetic ancestry that's actually found in the first Neolithic farmers that went from Anatolia and spread farming all across Europe. So actually it looks like they went in both directions to some extent. They had a much bigger effect in Europe, but they did try and go back into Africa. And that's just from one genome. You get these kind of really nice insights. Another paper which I'm on, but really I did nothing, But David Reich did all the work, and the other people. They were able to get 4 samples from the Shumlaka complex, archaeological complex, which is in the grass fields of Cameroon, a key place for understanding the Bantu expansion, because that's where we think the proto kind of Bantu languages come from. And they had 2 individuals that were 8,000 years old and 2 individuals that were 3,000 years old. So kind of either side of the Bantu expansion. And what was striking is these individuals genetically, 8,000 and 3,000 years, 5,000 years separated, were genetically pretty similar. Again, we have this kind of 5,000 years of genetic continuity almost in this small area. But they were very, very different from West Africans, Bantu speakers that live today. There's no, even the 3,000-year-old one. They were more like Central African hunter-gatherers. And this is the kind of insight we could only have gained from ancient DNA. We would never have known that this kind of diversity existed 3,000 years ago by looking at modern populations. We're blind to it unless we get ancient DNA. Similarly, we were just talking about pastoralism. I saw highlight a paper from another Stony Brook alumni, Elizabeth Sorchak, who's been really heavily involved in a lot of this ancient DNA work in Africa, where they were looking at, from Kenya, from Turkana Basin, a bunch of kind of pastoralists and pre-pastoralists, late Stone Age. And again, the remarkable thing you see is those individuals from the late Stone Age have a genetic kind of signal that looks a lot like Eastern African foragers and is very distinct from what would be the pastoralist genome, which suggests a big shift in ancestry and some kind of maybe migration and admixture. What's even more interesting is once you have this shift, from about 3,000 to 2,000, even a little bit later, there's pastoralist samples they have from a variety of different cultures, different pastoralist cultures. And though there's this incredible archaeological diversity about them, genetically, they're all pretty similar. And so that's a kind of interesting insight as well. It tells us about the demographics of that process. Cultural diversity, but actually not a kind of very small genetic kind of pool. Now, those have been interesting. I want to just draw attention to this paper by Brenna Henn, who was a faculty member here. I'm very sad that she left some years ago. This paper came out I think 2 weeks ago, big news in the New York Times. And it goes back to that first model that I came up with about San being the most divergent and in Pygmies and Niger-Cordophanians. As we've got more and more genetic data, it's been harder and harder to fit that model because we have now a lot more things that just don't seem to fit. So people have invoked things like archaic admixture from a ghost archaic hominin that goes in and maybe introgresses with West Africans, kind of mimicking what we see with Neanderthals, where we have actual DNA. But we still maintain that the first kind of divergence of human populations is 100, 200,000 years ago. It's just introgression from this ghost archaic hominin. What Brenner was able to show is actually with a much more complex model involving population structure that lasts for actually about 1 million years, with all these and this kind of loose connected gene flows, you can fit the data just as well, if not better. But it suggests a radically different kind of model. Then rather than thinking about trees, it's now a graph. It's a network of things. And it's really not about just bifurcations. It's coming together and things like that. The problem is if you're going to 1 million years for ancient DNA, we can't, these are all just made up populations. This is just models. We don't know anything about them. It would be great to have ancient DNA to even get some of these guys, but we don't know who they were. But if we could, it would be important. And as Brenner points out in the paper, other African populations, such as pre-Holocene ancient DNA samples, could further test our proposed models. We can get real empirical about this rather than these kind of guessing things. So this all leads to the natural conclusion of what is needed, which is a paleogenomics lab at the Turkana Basin Institute. And I will end there, because I'm pretty sure I went over time. Thank you.